Okay, so a centrosome is a structure that's going to really help pull the cell's genetic material to opposite sides of the cell. And a centrosome and a centriole are a little bit kind of tricky because a centriole is a part of a centrosome. So if you get confused about it, don't worry about it. For our purposes and our purposes only, let's think of a centriole and a centrosome as the same thing. I'm never going to ask you to differentiate them, but just kind of keep in mind in the back of your mind that when you hear both terms, that they're going to refer to structures. They're going to put out microtubules, specific mitotic spindles or spindle fibers where they're going to attach to the centromeres of chromosomes to pull them apart. And just so that it's super clear on how these things look, I'm kind of drawing a chromosome on, so this is a chromosome and this is a chromosome here. And that centromere is going to be here. Right, and that centromere is sort of this button that will sort of hold these two chromosomes together. And in fact, these two chromosomes that I've just sort of labeled in orange, each of these is going to be called sister chromatid. So this is a sister chromatid and that is a sister chromatid. So as I was referring to before, the centrosome is going to contain a pair of centrioles, right? So our purposes, I don't really want you to get too bogged down on. You could think of them as one as the same. But I want to, on the exams, I'm not going to put these two up to try to trick you or anything like that. The main thing is you knowing that what the role of these centrosomes or centrioles are for, and that's to pull the chromosome material apart. The asters are another structures that the centrioles put out. So centrioles put out both mitotic spindles slash spindle fibers. Sometimes we refer to them as both that are going to attach to the chromosome. And we have asters that are going to come from the back of the centriole to attach to the plasma membrane. All right, so mitosis is going to be divided into phases. So we have prophase, prometaphase, metaphase, anaphase, and telophase. And these stages are actually going to be continuous with one another, right? Just a little heads up, prophase and prometaphase we are going to talk about separately, but in some textbooks and even on the internet, depending on your source, prophase and metaphase may be wrapped up into one, right? So sometimes we're looking at them as a distinct event, but I do want to just point out the fact that some textbooks, including really some upper level textbooks, really combine pro-metaphase and pro-metaphase together. So we're not doing that, but I do want to just point that out. So with prophase, prophase kind of looks like this, right? So, and I didn't draw this super symmetrical, but that's okay. You're going to have your nucleus here, and you're going to have chromosomes, but they're going to be kind of condensing at this point. And you have your centrioles here, or centrosomes, and I'll just draw them as little cupcakes, right? And what they're going to do is actually go to their opposite pole, right? So this one, this one here would move here, and then this one here would move there, right? So, and at the end of this, you will have both of these centrosomes or centrioles at opposite ends of one another, right? Um, and the spindle fibers are going to appear. These spindle fibers are going to attach to the centromere, and the nuclear envelope is going to disappear. So if I were to show that, it would kind of look like this. So the nuclear envelope would then begin to sort of fragment and disassemble. The nucleolus, which you don't see at all, would have been disappeared as well. And then the chromosomes are actually going to condense, right? And they're going to, again, you have these sister chromatids held by a centromere. And again, if I were to draw it, it will sort of look like that. And with our little button there to hold these two sister chromatids together. And again, that button, again, is called a centromere. And I know it's a little bit confusing because we're talking about centromere, chromatid, chromosome. So there's a lot of C words here. All right, so this is from your book, and another heads up again. that some people combine prophase and prometaphase together. In fact, there are some people who combine early prophase, and prometaphase all together. So just keep that in mind for those who are studying maybe older textbooks or other textbooks. Okay, so in prophase, you're going to have the nuclear envelope, which is going to be here. It's going to start to fragment. The nucleolus is going to disappear, and the chromatin is going to start to condense. And if you notice here, guys, that the centrioles here are really not in the best location here, right? So you can see they're not at their opposite poles as they are here, right? So just wanted to point that out. Then you have the duplicated chromosomes. So the chromosomes have actually been duplicated, and the you can see that there is no nucleus, right? In fact, these little purple fragments here that I'm sort of clicking on are sort of the remnants of that. And in prometaphase, you see the centrioles are on opposite poles, and now you have these spindle fibers that are coming out, right? The asters are going to be going toward the back. So these are going to be like your asters kind of attaching here. So spindle fibers go forward, asters go back, right? All right, so as I just talked about before, is that the spindle fibers are going to attach to the centromeres, and the chromosomes continue to shorten and thicken, right? So, and then what happens next is that the chromosomes are going to be randomly placed in the nucleus. And put a pin in this because this is going to be important when we talk about um, meiosis. Metaphase, so again, the metaphase plate is just down a perpendicular axis. So just so that it's clear to you, a metaphase plate could be this. This is how you're normally, this is probably most likely going to be seeing it. Or it can be this. So just so that it's clear, right? It can't be sort of, it cannot be like sideways like this, right? That would be really funny. But so just make sure I just kind of put X. It can't be that way. It's either going to be this way or this way. But anyway, so that is going to be your metaphase plate here. And chrome and during metaphase they're going to line up that way. And just keep in mind, guys, it's good for you to keep your notes in front of you. I can't go back in the way that I'm recording this, so I can only move forward. Okay, so this is our metaphase, and you can see that these chromosomes are going to be lined up on the equator. And always think of metaphase middle, like middle metaphase, right? It kind of sounds with the two M's. And for anaphase, I always think of these sister chromatids are being pulled apart, right? So always think of like apart, like apart anaphase, right? So hopefully maybe that helps you. And then the telophase is really where you're going to have um, the nuclear envelope reforming, right? And the chromosomes are going to be right in their nucleus. And really, telophase really is at the heart of what mitosis is about. The whole purpose of mitosis is to separate that genetic material, right? So by separating that genetic material, now you can move on to cytokinesis, which is the separation of that a cytoplasmic material, right? So just kind of keep in mind that. And the telltale sign of telophase is that cleavage furrow, right? So that cleavage furrow is really a good sign there. All right, so anaphase is when these centromeres are going to divide, and really, and I like to think of just being split apart. And the sister chromatids are going to separate, and they're going to move to their opposite poles, right? And now, once these sister chromatids are separated, we call them chromosomes. So there's a lot. That's the interesting thing about biology, is that they kind of have different names for different things, even if it's just one little change, right? A good example would be like a uterus and a womb, right? A uterus is a uterus. When there's a baby in it, it's a womb, right? So just kind of keep in mind that there may be changes in words and word meanings just based on a loss or gain of something, right? So these sister chromatids, once they are pulled apart, their new name is a chromosome. Telophase is going to begin when the chromosomes arrive at their opposite poles. The chromosomes are going to really now begin to unwind and become chromatin again. The spindles from The centrosomes or centrioles are going to disappear. The nuclear envelope is actually going to reappear. The nucleolus is going to reappear as well. And again, I think we talked about this before. We're not going to talk about it too much, but the nucleolus is involved in making ribosomes. And telophase is characterized by the presence of two daughter nuclei, which again is really the hallmark of what mitosis is about, right? Mitosis is about that division of the nuclear material, right? And telophase is really that button or that bow at the end uh, of that, a kind of a punctuation. So cytokinesis is going to be the division of the cytoplasm in the organelles, right? So this contrasts with mitosis, right? Mitosis is the division of the nuclear material. Cytokinesis is going to be division of the cytoplasm and organelles. Please keep in mind that mitosis and cytokinesis are not the same. Though cytokinesis follows mitosis, mitosis happens first, cytokinesis happens last, right? So please keep that in mind that they are different and they have different roles, right? Remember that mitosis is a division of the genetic material, while cytokinesis is a division of the cell and all of its liquid and goo and organelles. Okay, and this cleavage furrow that you saw in telophase, that's going to sort of invaginate so much that it actually separates completely. Okay, all right. So meiosis is the division of sex cells, right? So this is if you're a woman, this is going to be ovaries. I'm sorry, your ovums. And if you're a man, this is going to be the division of spermatozoa, right? Remember, ovums are going to be made by your ovaries, spermatozoa or sperm are going to be made by the testes. And so the meiosis is really that particular story there. Okay, so this is the one main thing of meiosis compared to mitosis is that it's going to result in four daughter cells, right? Each with each having into one type of chromosome, right? So a parent cell is going to be 2N, while the daughter cell is going to be N. Okay, one of the most important things about meiosis is that it's actually going to introduce genetic variation. And this is going to make it so that each of the daughter cell is actually different from the parent cell. So, and this allows for new combinations of genetic material. Okay, so at the start of meiosis, each parent cell is going to be diploid. So a diploid just means it has two copies. Now, N and for humans should be 23, right? So this is just a mathematical expression. So 2 times 23 would be 46 chromosomes, and that's actually what you have. So what this is saying is that each sperm and each ovum is going to have 23 chromosomes. Therefore, when they come together, you're going to make an individual that has 46 chromosomes. This is kind of looking at the picture from your book, and you can see that during for meiosis, you have replication of the genetic material, and during replication, what you have is each of the chromosome making a copy of itself, right? So you end up actually having 92 chromosomes before meiosis 1. And at the end here, you're going to have 46 chromosomes, right? And then at the beginning of meiosis 2, this is going to be whittled down to 23 by the end of meiosis 2, right? So you're going to end up with 23 so I'm sorry, 23 chromosomes. Okay, and we're going to talk about synapses. Synapses is just this process in which these chromosomes line up, right? So, and you do not have this in mitosis, right? You have that lining up, but you have chromosome 1 from your mom, chromosome 2 from your dad, and they actually line up together, and this is referred to as synapses, right? This does not happen in mitosis because just all of them are just duplicated, and then each one gets a copy, right? So it's a little bit different. Okay, so during meiosis and meiosis 1 and 2, all the DNA is going to be replicated prior to meiosis 1. Synapsis is when you have homologous chromosomes are going to come together. So remember, homologous chromosomes are going to be, this is really the same term we use for mitosis, except instead in mitosis we call these sister chromatids. So again, the names kind of switch up depending on whether you're referring to mitosis or meiosis. And you're going to line up on the metaphase plate during meiosis one, and this is going to keep the chromatids close during the very first first two phases of meiosis one. And remember, you're going to have four chromatids because you're going to have you're going to say what happens is that you have a chromosome from mom, so we'll put this one for mom, and then you have a chromosome from dad, right? And we'll do it this way. And then what happens in meiosis one is that you actually make a copy, right? So you actually make a copy of this, right? And so what you have now are four chromatids, right? One, two, three, four, right? And so that's actually what's happening in meiosis one, right? So there is a pause during for meiosis one and meiosis two. There is no replication, and this pause between meiosis one and meiosis two is called interkinesis, right? Sort of like a little pause, like an intermission, like for a very long movie or play. All right, so during meiosis two, the centromeres are going to divide or separate, and they're going to then pull toward the opposite poles. And now, once the sister chromatids separate, they're going to be called chromosomes. So again, they do have these different names depending on just a little bit of function and appearance of where they're at. Um, and the daughter cells that come off of these are going to be called gametes, and the gametes that exist are going to be sperm and ovums, right? So, and when we have a fertilization, the fertilization is going to restore the diploid number of chromosomes in the zygote. So remember, we are 46 chromosomes, but I'll just draw an ovum here, and then I can draw like a sperm, and this is 23. So when the sperm fertilizes this ovum, you end up with a person that has 46. And that's somebody, right? Okay. All right. So I can't zoom in the way that I am trying to record this, but I hope that you can look in your book or if you have the notes in front of you. But in meiosis one, you're going to have a prophase in which these homologous chromosomes are going to pair up for synapses. So again, what happens is during this whole process is that you've made a copy of your mom's chromosome and your dad's chromosome, and because you made a copy of it, you have four chromatids or sister chromatids, and now they're going to come together during synapsis. And then they're also going to cross over, and we'll talk a little bit about that later. And then you have metaphase one, in which these chromosomes will line up randomly. And this is called independent assortment. Sometimes we call this independent alignment. And again, we're going to talk about this in a moment. And then anaphase one is going to be when these chromosomes are actually being pulled apart. The homologous chromosomes are being pulled apart. And then telophase is when the daughter cells are going to have one chromosome from each of the homologous pair, right? So, here in interkinesis, this is nothing has really changed. There's just sort of a pause between meiosis one and meiosis two, which we're going to be talking about shortly. All right, meiosis two. And again, but both of these are analogous, right? So this side and this side are the same. I'm just going to kind of draw on this side here. But what occurs in prophase two is that, again, each cell is going to have one chromosome from each of the homologous pair, and then these chromosomes are going to line up on the metaphase plate. So you can see that here, right? So meiosis two looks very similar to mitosis. So if you're like, it looks like mitosis, you're not crazy. And then and then again, anaphase two chromosomes being pulled apart, and then telophase is when each cell is going to have its own complement, right? And cytokinesis is going to occur in which you're going to make two daughter cells. And because the same process is occurring on this side, you end up with four of these haploid cells, right? So you end up with a 46 here, and then you end up with a 23 here, right? And so if when you mate or have a kid, your 23 is matched up with your partner's 23, and then you make a 46 chromosome person. Yep. So the purpose of meiosis is to ensure genetic variation, right? And so it ensures that the offspring will be diploid and have the combination of genetic characteristics from each parent, right? So what's happening is that your cells are taking genetic material from your mom and your dad and sort of mixing them into a pot, right? And we'll look at that in just a moment, right? Um, and both meiosis 1 and 2 have the same four stages as the nuclear division of mitosis rather. So it should look a little bit familiar. All right, so the main important thing in meiosis is synapses and crossing over. Crossing over is huge, right? So synapses is those homologous chromosomes lining up, right? Remember, you're going to have chromosomes from your mom and your dad, and what's going to end up happening is that they make a copy of your mom chromosome and your dad chromosome, okay? And then they're going to actually line up side by side. So, and I can actually kind of draw that. Wait a minute. And it will kind of look like this. Yeah, and this is really synapses, right? This is these chromosomes lining up side by side, and then crossing over is super important. Wink, 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 because this is going to exchange genetic material between the non-sister chromatids of the homologous pair. So this is a copy of your chromosome 1, right? Chromosome 1 from mom, chromosome 1 from dad, right? And this is kind of making so you're figuring out like what is a chromosome 1 you are going to pass on to your children, right? So that's really the purpose of crossing. Okay, so synapses and crossing over is very important. I like this slide, though it's a little funky. In red here, this is, let's say, it's from the mom, and in blue, this is from the dad. So the mom, you can see, is big A, big A, big B, big C, big D, and the father is little a, little bit, little b, little c, little d. And you can see that they're homologous. In fact, let's just say they're chromosome 1 for mom and dad, right? This is your first chromosome for mom and dad, and mom has the dominant traits and dad has the recessive traits. And when we look at this, what you get from your mom is this 1, and then dad, the original from your dad, is 3. And what you're doing in 2 and 4 is that you're making a copy, right? So now because you made a copy, 1 and 2 are sort of a pair, and 3 or 4 is sort of a pair because this is the 4, it represents a copy of the third chromosome. And so now you're going to line these up together. And so that's synapses, right? And synapses is basically lining up these homologous chromosomes from your mom and your dad. Okay, um, and crossing over, this is really cool because what you see is that it's showing you crossing over between 1 and 3. I think they mean chromosome 2 and 3, but I think that's an error. But in any case, this is chromosome 1 and this is chromosome 3, and you can see that they're now crossing over. I don't know if you can see that, right? So you can see it's crossing over, and the resulting chromosomes, you can see this is why I think it's an error, is because the ones that are only changed are going to be 2 and 3, right? So you can see that like if this was a gametes from your mom and your sperm, one child, for example, would get exact copy from your receive one copy, the exact copy you got from your mom. 2 and 3 would receive a kind of copies that are sort of amalgamation or a mixture of your mom and dad. And the fourth copy here would just be straight up from your dad, right? And this same process, guys, is actually going to happen uh, in um, in your children, right? So it's actually really, really cool. So anyway, hopefully it isn't too confusing. It's super important, but synapses and crossing over increase variability in your gametes, in your sperm and ovums. Okay. Um, The other thing that happens is going to be in metaphase is going to be independent assortment. Sometimes we call this independent alignment. And this refers to when homologous pairs are going to be aligned independently at the equator. So if you look at this picture here, you can actually see that the chromosomes, you can see like blue, blue, blue here. And on this side, you have blue, blue, red. And on this side, you have blue, red, blue, and it goes on and on and on, right? And this independent assortment during cell division or meiosis actually ensures that you have variability in the gametes that are being produced. So in spermatogenesis and oogenesis, these are specific terms that happen in men and women. So meiosis is going to be part of spermatogenesis. Spermatogenesis is going to be the division of sperm, and this is going to occur in the male. And oogenesis, which is a kind of a funny word, is going to be the production of ovums in females, right? So following meiosis, the daughter cells that are being produced mature into gametes, right? And we'll talk a little bit about that process in the next few slides. All right, so spermatogenesis is going to occur continually in the testes starting at puberty, and men produce over 300,000 sperm per minute, which is about 400 million sperm per day, right? And how this works is that sperm go through this um, transition where it goes from primary spermatocyte, secondary spermatocyte, spermatid, and then finally sperm, right? There is sort of a lineage that or maturation that these sperm go through that we'll look at. Okay. And as I just said before, the primary spermatocytes are going to turn into secondary spermatocytes, uh, and then um, and then during meiosis two, those secondary spermatocytes are going to turn into four spermatids, right? You want to kind of take a look at that in the book if that's not too clear, or see me during office hours. And then these spermatids are going to mature into sperm or spermatozoa, right? That's how I referred to before. You have primary spermatocytes, secondary spermatocytes, spermatids, and then that goes into sperm or spermatozoa, right? In males, all four of the daughter cells actually become sperm. In females, this is actually not the case. And when we get into female reproduction, we'll kind of dig into that a little bit more. Okay, so this is a kind of a picture here. And this big, large one, if you're like, what the heck is that? You don't need to know. That's actually a spermatogonia. So that's sort of a stem cell that men have in their testes that then generate sperm. But to get to the picture, you have meiosis one, and during meiosis one, you're going to actually kind of start off with 46 chromosomes. That's going to double to 92, and then at the end you're going to have 46, right? And these are going to be really haploid, right? They're going to really just have one copy. Actually, I'm so sorry, not they're going to be they're going to be haploid. Actually, that is right. They're going to be haploid here, and they're going to have two copies, so it's going to have 46. So it's going to actually have 46 chromosomes at the end of meiosis one. And at meiosis two, these 46 chromosomes are actually going to turn into four 23. So you have a 23 here, 23 here, 23 here, 23 here, 23 here. And what's happening is that these one is turning into two. So you're for spermatids to sperm is more of a morphological change, right? These spermatids sort of wiggle and wiggle, and they actually make a midpiece and a flagella, and they make an acrosome. These are all components of the sperm. And once they have matured, they go from the testes to the epididymis, where they actually finish their maturing. There's actually more maturation that occurs, but that's neither here or there. All right, so let's go to oogenesis. So oogenesis is going to be referring to the development of ovums in the ovary, right? Um, the difference between the spermatogenesis and oogenesis is that women, when they're born, they have all of the ovums that they have. And in fact, these ovums are actually called primary oocytes, and they're going to be arrested in that stage of prophase one. And at this stage, these primary oocytes are actually going to be diploid, and when they divide during meiosis one, they're going to turn into two haploid cells. And remember that for oogenesis, there's only one that's produced. So the one that isn't going to be the cell that isn't going to be divide, they actually degenerate, or they actually just are put out as polar bodies. So these polar bodies are really discarded chromosomes. Okay, but in any case, the primary oocyte is actually going to turn into the secondary oocyte, and that's going to receive almost all of the cytoplasm. And the one that loses, in a sense, it turns into a polar body. So it's kind of interesting. And these polar bodies, just think of them as just sort of waste products that for in ovums, that they kind of get rid of their chromosomes, right? So they do it in men. The chromosomes are sort of are going to be distributed to all four of the cells. In women, or in oogenesis, this only happens in one. Okay, so a picture sometimes is worth 1,000 words, right? So in meiosis one, you can actually see that this ovum, the ovum, it has divided, but it has like a polar body, and this is just again a waste product, right? In meiosis two, what happens is this only occurs when fertilization occurs. So when fertilization occurs, meiosis two is on and popping, right? So what occurs in meiosis two, um, is if meiosis two completes, and then a second polar body is formed. So again, these polar bodies are again how ovums discard their unused chromosomes, right? Because you don't want these unused chromosomes causing a chromosomal abnormality, right? So you want to make sure that you have 23 chromosomes for mom and dad. Okay, and so here at fertilization, you have a distinct individual, right? You have 46, which is actually really cool, right? Because here you have 23, here you have 23, here you have 23, here you have 23, and when they come together, you make a distinct individual that is a mixture of both the sperm's DNA, and the female's DNA, or the ovum's DNA, which is in the nucleus there. Okay, so the secondary oocyte is going to leave the ovary during ovulation, and this is going to enter the uterine tube. So another name for the uterine tube is called the fallopian tube, right? And so if the ovum is fertilized, the oocyte is going to be activated to complete that second meiotic division. So because what happens is that typically ovums are going to be arrested at a metaphase of meiosis two, right? And the reason is, and it's actually kind of funny, because what occurs is that all of the chromosomes in the ovum are sort of lined up in the middle, and they're literally just patiently waiting for fertilization by the sperm, in which the sperm will then orient its chromosomes in the middle of that cell and begin the process of fertilization, right? So, but in any case, so following meiosis two, there's going to be one ovum, and two or possibly three polar bodies. And again, these polar bodies are just simply discarded chromosomes. And then you want to have polar bodies because you want to discard unnecessary chromosomes while keeping most of the cytoplasm in the ovum itself. So the significance of meiosis is, it cannot be overstated. The function is to keep the chromosomal number constant from generation to generation, right? You don't want one person to have 49 chromosomes and another person to have 52. You want to have everyone to have 46. So that's a biggie. The other one is genetic recombination, right? And one is that you're making the significance of meiosis is to ensure that the offspring will be genetically different from each other and the parents, right? The other is going to result from crossing over and independent alignment of chromosomes, right? And so, again, crossing over is when you're mixing your chromosome 1 from mom and mixing chromosome 2 from dad and giving that as to your offspring, right? So that really is really increasing that diversity that you're offering, that you're giving to your children. And this diversity is actually needed to survive in changing conditions, right? The most common example will be sickle cell anemia and malaria. Sickle cell anemia it's a debilitating syndrome in which people have pain. It can kill you, and because of the red blood cells have been sickled, they kind of like a sickled shape rather than sort of that oval or circular shape. And the reason sickle cell anemia developed is really a protection against malaria, right? So in areas where there's high malarial infestation, sickle cell anemia is actually a very good survival strategy, right? It allows the next generation to live, even though it may cut the entire lifespan of the person who has it. Okay. So anyway, you don't need to worry about sickle cell anemia right now, but I did want to just really touch home on why having diversity in genes is important. It also helps in immunity too, but again, I don't really want to get too far into the weeds. So I love this slide. This is a comparison of meiosis and mitosis. Unfortunately, I can't zoom in like I like to do. If I zoom in, the program gets crazy. But I do want to just point out that in mitosis, which we've already talked about, there is no synapses. The chromosomes will line up along the metaphase plate. You have anaphase, which are going to separate those chromatids together, and the telophase, which are going to daughter cells are going to form. And in fact, there is a lot of similarity between the end of meiosis one and the end of mitosis. And in fact, what ends up happening during cytokinesis, you end up with two cells, right? With at the end of mitosis, you end up with four. But at the end of meiosis, these two are really synonymous because what occurs is that the end product of telophase one will then go into meiosis two, and then you will end up these two daughter cells will make four, right? So you end up with four. So that's really the big upshot of it is that meiosis is going to end making four, which is going to be n, right? Just 23, and here you're going to have two, which is going to be 2n, right? So and essentially, if you don't like that, just think of it as 46 chromosomes here, and you're going to have 4 23s here, two 46s, two from 4 23s. But anyway, all right, so what I wanted to just hit on is just synapses and crossing over. Remember that synapses is when your chromosomes, your homologous chromosomes are going to line up. So chromosome 1 from mom, chromosome 2 from dad, they come together, they duplicate, and because these two chromosomes duplicate, you have four chromatids. They kind of come together, and that coming together is called synapses. You also have crossing over. Crossing over is when these duplicated chromosomes from mom and dad, they sort of intermingle, right? And that intermingling is referred to as crossing over. And then the third thing that happens in meiosis that you don't see in mitosis is going to be these independent assortment or independent alignment, in which these chromosomes will line up differently on the metaphase plate, meaning that each daughter cell is going to receive a different complement of the chromosomes. Okay, all right. So, and we're not going to get too much into the weeds here because we're going to talk more about chromosomal inheritance itself at a later lecture. But generally, humans are going to receive 22 pairs of autosomes and then two sex chromosomes. And these two sex chromosomes are going to be really going to be the what we consider the 23rd pair. And a 23rd pair or 23 chromosome is going to be XX if you're a girl, or it's going to be XY if you're a guy. Okay. All right. So, and so each pair of the autosomes, 22, are going to carry alleles for a particular trait, like blue eyes, curly hair, blonde hair, so on and so forth, right? And alleles can be different. So, in other words, you can have an allele from one of the chromosomes. Let's say you have one from your mom, and you have another chromosome from your dad, right? And say one calls for freckles, but the other one does not, right? And so we are not going to get into the weeds on what happens there, it's like dominant and recessive, but I do want to just put a little foreshadowing there for a future in the future. Okay, so now I just want to talk a little bit about chromosomal structure, about how environmental agents or environmental contaminants like radiation, organic chemicals, viruses, even bacteria, being exposed to carcinogens, for example, can all cause chromosomes to break or mutate, right? So ordinarily, when you have a break on a chromosome, those two ends of the chromosomes will actually reunite. But sometimes broken ends do not rejoin as the same pattern as they were before, and that can lead to disease or derangements in the functions of a particular cell. Okay, so the changes in the chromosomal structure are going to include deletions, duplications, and inversions, right? We will not be talking about translocations. It's not that I have anything against it. I just don't think it's necessarily, we have a lot to cover, so we're not going to cover it. So you just need to know these three. Um, so a deletion is probably what is very easy, I think, conceptually to understand, is when a part of a chromosome breaks off or is not included anymore. So it's gone, right? And so that typically when you have a deletion, you typically have some type of a disease, right? Or some type of abnormality that's going to occur. A duplication is when a chromosome segment appears more than once on a chromosome. So this a part of a chromosome has been sort of duplicated by accident, right? And sometimes it's caused an issue, sometimes it doesn't. Many times it causes an issue, especially because you could be making more of a particular protein or more of a particular gene that could be helpful, or it's which you may have a positive benefit, or it may be deleterious in which it could be you would have some abnormal effect, right? So a duplication is sort of a wild card whether it will help or hurt the person who has a duplicated chromosome. An inversion is a segment in which a chromosome is inverted, right? And so this usually doesn't cause much of a problem, though it can, because at least all the genes are present, right? So hey, it's backwards, but at least it's actually there. Okay, so these are pictures from your book. Actually, like it. Sometimes a picture is worth a thousand words. And so a deletion. So here you have A, and then when you have a deletion, you see that A is actually fallen off. So you don't have A here anymore. In duplication, you see that you have a CDF, and then here you have CD, CD, FG. So in other words, this CD has actually been duplicated twice, right? So sometimes it can cause problems, sometimes it won't. Sometimes there's a benefit, sometimes there isn't, sometimes there is a bad thing that happens. So it could be literally good, bad, or neutral depending on what actually is being duplicated. Okay. An inversion here typically, it doesn't really matter whether you have A, B, C, D going or A, B, D, C, because the genes are actually already there, and it's for the most part, this typically won't cause disease, right? Though it's not. We're not going to go into the exceptions. I just want to be very, very general here, right? And again, we are not talking about translocations in this class. Okay, so let's move on. Okay, so some human syndromes. So Williams syndrome is when chromosome 7 loses a piece, and so you can have like a turned up nose, wide mouth, small chin, large ears, and even though they have poor academic skills, they these individuals tend to have excellent verbal and musical abilities, right? So the gene for elastin is missing, so this affects the cardiovascular system and causes the skin to age prematurely. So because there's probably a lot of different genes on, there's probably lots and lots of genes that are being lost on chromosome 7, right? So it probably affects different things. All right, so let's go here. And again, this is just your deletion, just kind of showing you the deletion. You can see that you can have the chin, the turned up nose. I don't know if you can see the nose there, and then the ears there, right? So, and again, this is just to show you. I promise you, I'm not going to show a picture of show a picture of this and say, okay, what is this? But I could show a picture of this, right, and ask you what type of chromosomal defect is this, right? So, all right, let's move on. All right, so this is cry du chat. So as you, if I'm correct, I think this is a French word, which I think means cry of the cat, and the reason they call them that is because the way that their larynx and glottis, which is in the voice box, are is that when they cry, it actually kind of sounds like a cat, right? So these individuals may have a small head, they may have some intellectual disabilities and some facial abnormalities.